Okay. So first, I would like to thank very much Josiane for her kind invitation and for giving me the opportunity to present the work of my group. For several years, our research has been focusing on protein maintenance either by degradation or by repair. And in my group, we are mainly interested in methionine sulfoxide reductases, which are oxidized repair enzymes, and we are focused on their involvement in aging and in protection against oxidative damage. So I'm going to present what we did during over the last few years and also more recently. So, yes. Proteins are very susceptible to almost all forms of reactive oxygen species, mainly produced by the mitochondria, and oxidation of proteins lead to the formation of carbonyl groups in proteins, or sulfonic acids, or even cleavage of polypeptide chain that are this modification being irreversible. So this kind of oxidation of this protein oxidation has to be eliminated by degradation. So these proteins are degraded by the proteasome in cytoplasm and nucleus and by the long protease in mitochondria. On the other hand, some other modifications that affecting sulfur amino acids can be repaired, so are reversible. For example, methionine sulfoxide reductase, which is the oxidized product of methionine, can be repaired by methionine sulfoxide reductases. And we know that, unfortunately, these protein maintenance systems are no longer efficient with age, and we know that proteasome and long protease activities are declining with age, and we just listened to a very nice talk about it from Kelvin Davis and Statis Gonos. And almost 10 years ago, we showed in the lab that the repair enzymes methionine sulfoxide reductase are decreasing with age, hence contributing to the accumulation of age-related oxidized proteins. So methionine is oxidized in two diastereoisomers of methionine sulfoxide, and in proteins, methionine S-sulfoxide can be reversed by MSRA, and methionine R-sulfoxide can be reversed back by MSRB. The two enzymes function with thioredoxin reductase and NADPH to reactivate the active site, the active cysteine. So the methionine sulfoxide reductase system is very important considering its remarkable conservation during evolution and its subcellular localization. In mammals, MSRA is encoded by one gene and is expressed in cytosol and mitochondria and also nucleus. On the other hand, MSRB proteins are encoded by three different genes, MSRB1 is expressed in cytosol and nucleus. MSRB2 is located in mitochondria, and MSRB3 and B and A, generated by alternative splicing, are located in reticulum, endoplasmic reticulum, and mitochondria, respectively. So all the MSR contain a catalytic cysteine, and some of them contains one recycling or two recycling cysteine. For MSRB1, this is selenocysteine instead of cysteine. And all MSRB are zinc-binding proteins, and zinc is very important for their folding and their activities. So, in addition to its repair role, the MSR system has been involved in redox homeostasis regulations by permitting the cyclic oxidation reduction of methionine, of exposed methionines in some proteins. And that has been described as a very important antioxidant system, and therefore MSRA has been, is believed to have an important role in reactive oxygen species scavenging and in protection of cells against oxidative stress, and also in longevity. In the lab, we have overexpressed MSRA in W838 fibroblasts, and we showed that MSRA targeted in both mitochondria and cytosol can induce the MSR activity, and of course, and protect cells against oxidative stress mediated by H2O2. And this protection is due not only by the decrease of reactive oxygen species in MSRA overexpressing cells compared to control cells, but also by prevention of accumulation of oxidized protein, as you can see in this oxyblot, in which you can see an increase of oxidized protein in MSRA cells with H2O2, but this increase is is less important compared to control cells. So the protection against protein oxidative damage and the decrease of reactive oxygen species levels can explain the protection against H2O2 we observe in these cells. As far as MSRB is concerned, little is known about its involvement in protection against oxidative stress and longevity. So we have overexpressed MSRB2. We have chosen MSRB2 because MSRB2, if you remember, is restricted to mitochondria. And MSRB2 being targeted to mitochondria, it can be very important in the mitochondrial protein maintenance. So we have overexpressed MSRB2 in human leukemia cell line multifor, and very interestingly, this cell line is one of the very few cell lines that doesn't contain neither MSRA protein, neither the gene, neither MSRA protein and MSRB3. So we showed that the overexpression of MSRB2 in mitochondria lead to the increase of MSR activity and to a better resistance of cells against oxidative stress. The overexpression MSRB2 clearly protects against H2O2 mediated cell death by delaying both necrosis and apoptosis, as you can see in this blot, where you can see the inhibition of PARP1 cleavage. The depolarization of the mitochondrial membrane is less important in MSRB2 provoked. So this depolarization was provoked by oxidative stress, but H2O2, and is less important in MSRB2 overexpressing cells compared to control cells. As an example, for treatment of 75 micromolar of H2O2, you can see that the green fluorescence signal of GC1 is increased in control cells compared to MSRB2 overexpressing cells that retains almost the same fluorescence. And in control cells, this modification of mitochondrial potential membrane lead to the production of reactive oxygen species, the increase of reactive oxygen species production. In contrast, in MSRB2 overexpressing cells, the maintenance of this potential membrane leads to a decrease of reactive oxygen species production. So, when we look at the oxidized protein, we can see that MSRB2 overexpression can protect cells against accumulation of oxidatively modified proteins, of carbonylated proteins. And we know that the oxidized protein in homeostasis conditions are degraded by the proteasome. But in stress condition, in some stress condition, because we saw that in certain stress condition proteasome can be activated, but in other stress condition, when in other stress condition, yes, we can talk about it. In other stress conditions, so the proteasome can be altered in activity and also content. So we show that when we treated the cells with H2O2, we have in control cells a decrease of proteasome activity without modulation of its expression, and this decline can be limited when in cells that overexpress MSRB2, indicating that MSRB2 can protect proteasome activity during stress condition, in stress situation. But by using proteasome inhibitor, we showed that the amount of oxidatively modified proteins is still lower in MSRB2 overexpressing cells compared to control cells, even if the proteasome was inhibited, indicating that not only the protection of the proteasome activity can explain the protection against the prevention of the accumulation of oxidized protein, but also the antioxidant properties of MSRB2 contributes to this prevention. So to sum up what we found in this first part, we found that MSR overexpression can lead to the decrease of reactive oxygen species and the protection of proteasome that can decrease the level of oxidized proteins in cells. We also found that in MSRB2 overexpressing cells, we have a preservation of mitochondrial membrane, so preservation of mitochondrial integrity and a decrease of apoptosis. And we also showed in another study that I have no time to detail now, that MSR overexpression can modulate the expression of some proteins, and especially some proteins are upregulated. These proteins are involved in redox homeostasis and protein folding, and some other proteins are downregulated, expressed, and these proteins are involved in apoptosis. So, altogether, our results indicate that protein repair can be an important, very important part of protein homeostasis during aging or during oxidative stress, and that MSR can be a very interesting target, anti-aging target for cosmetic company. And since MSR is expressed at very high level in the skin and is decreased with aging and after UV exposition. So in collaboration with Christian Dior Cosmetic, we have we have found a compound called aminolumine, and this compound is the first ingredient capable of deoxidizing proteins. And since it increases the activity of MSR and decreases the oxidized protein level. Okay, now the study I would like to talk now has been initiated in the frame of the European Zinkage project. And zinc is a trace element involved in antioxidant defense and maintenance of immunitary functions. And during aging, a combination of reduced intake, malabsorption, and sequestration by metallothionein decrease the concentration of zinc, leading to the alteration of cellular homeostasis, especially in the immune system. So, the aim of this study was to determine the impact of zinc on protein maintenance system activities, that is, proteasome and MSR, in lymphocytes of elderly. In fact, proteasome, we showed previously in the lab that proteasome activity is decreasing in lymphocytes of old people during aging, and I just show you that MSR is very important in oxidative protection. And if you remember, MSRB proteins are zinc-binding proteins, and zinc is very important for the activity of MSR. So we first analyzed this effect on cohort studies of elderly from 59 to 85 years old, sorry. And these cohorts of elderly were recruited by our collaborator from Italy and Poland, and we showed that 7 weeks of supplementation can increase the plasmatic zinc levels in these people, and also can lead to a decrease of oxidized protein level in lymphocytes. That can be explained by an increase in lymphocytes of proteasome activity in people supplemented with zinc compared to control, and to an increase of MSR activity. So in these people, zinc can activate protein maintenance system, proteasome and MSR. And to better understand, to elucidate the role of zinc in MSR system, we treated the cells, so multiple cells, control cells or MSRA and or MSRB overexpressing cells with zinc during 24 hours. And we can show here that zinc increases the activity of MSR only in control cells, and this effect is less evident in MSR overexpressing cells. So this activation, so this is an activation of MSR without modulation of its expression, in contrast to methylthionein, as you can see here by PCR and also by Western blot. So zinc has also a pro-oxidant effect, and we showed that zinc increases the reactive oxygen species production, decrease the viability, so increase the cell death, and increase protein oxidation. In contrast, when we treat our cells, the MSR overexpressing cells, we can see that these cells, clearly the overexpression of MSRA or MSRB2 limit the pro-oxidant effect of zinc. As you can see here, there are less production of reactive oxygen species, less cell death, and less accumulation of protein oxidation. So, zinc has a dual effect on our cells. In one hand, it has an antioxidant effect by induction of antioxidative proteins such as the metallothioneins and methionine sulfoxide reductases. And it has also a pro-oxidant effect by the production, by inducing the reactive oxygen species production and protein oxidation and cell death. And in controlled cells, the pro-oxidant is more important than antioxidant effect, whereas in the cells overexpressing MSRB or MSRA, the antioxidant effect is more important than the pro-oxidant effect. So it's difficult to explain, but we propose that in these cells, the MSRA or MSRB2 overexpression confers to the cells the capacity to fight reactive oxygen species and allows the zinc to the antioxidant effect of zinc to take place. Okay, now just an introduction of what we are doing now. Our ongoing studies aim to elucidate the differential role of the different MSR. And so for this purpose, we used RNA strategy to inhibit one MSR, to inhibit MSRA or one MSRB or both. And here it's an example with MSRA and MSRB1. So we inhibit MSRA, MSRB1, or both in cells, in human embryonic kidney cells. And you can see that if we reduce the expression of MSR, we have a reduction of residual MSR activity, and this activity is no longer detectable in the double mutants. So we use these cells in 2D Dye experiments that allows the identification of differentially expressed protein in different situations. So our aim is to compare the protein profile from control cells to MSRA the control cell to cells inhibited for MSRA or cells inhibited for MSRB or cells inhibited for both in basal condition and stress condition. Mediated by taurine chloramine. And so we have done 10 statistical analyses, and the proteins which are differentially expressed were analyzed by mass spectrometry, liquid chromatography and mass spectrometry, and the identified proteins mostly belong to cytoskeletal proteins, to redox homeostasis, and also transcription traduction. And you can see here in these diagrams that that the double mutants are more susceptible to modifications, to protein modification, to protein modulation, and that some proteins are upregulated and some others are downregulated. And if you want more information, I invite you to go to see the poster number 30, the poster to talk with Sabrina Rajay, which has the poster number 30. And maybe it's not too late to say, vote for her, please. Okay, so now I would like to thank the people who participate in this work. Of course, Bertrand Friguet, which is the head of the laboratory, the old MSR team, which is not old in age, but they are no longer in the lab, the young MSR team, they are young and they are in the lab. Our partners from LVMH, Christian Dior, and from the European project. And thank you very much for your attention. So, just to finish, our lab is in this building, which is not so nice at the Centre des Cordeliers, but we are in front of the Seine and very near from Notre Dame. Thank you.
This question is open.
It's late.
Fundamentally from Renzo. You mentioned that FMSR was particularly presenting scheme.
Yes.
Could you precise on more parameters with crisis situation?
Okay. So MSR is highly expressed in keratinocytes and also in melanocytes and in the epidermis and in fibroblasts in the dermis. But the expression in fibroblasts is less important than in epidermis.
And does it decrease with age?
Yes.
In keratinocytes especially?
In keratinocytes especially, I'm not sure. In fibroblasts, yes. In keratinocytes especially, I'm not sure. I'm not sure. No, I'm not sure. We have done the experiment after UV exposition, but not with age. Yes.
So I don't think you said there was I don't think there's a problem in terms of how we saw an increase in activity and you saw a decrease. In the model we were looking at is specifically an adaptive
yes,
or hormetic model. So we specifically chose concentrations of peroxide and timings that gave us an increase.
Yes, and there is also
looking for toxic effects. But could I ask how you measured proteasome in your study?
Sorry?
Could I ask how you measured proteasome activity in the study you did?
Yes, we measure with fluorogenic peptides. Yes, but this is a question, I think, of time of recovery too, because no.
I don't know. I think you were looking for toxic effect. We were specifically looking for adaptations.
Yes, of course.